Okay, so this is chapter 1, the main themes of microbiology, part 7, the final section of this chapter, naming, classifying, and identifying microorganisms. So in this section, we'll learn about how to differentiate among nomenclature, taxonomy, and classification. And again, that's just some terminology, understanding what those things mean, and create a mnemonic device for remembering categories, taxonomic categories, write the binomial names for a microorganism, be able to diagram the three major domains, and explain the difference between traditional and molecular approaches to taxonomy. So, what is naming, classification, and identifying microorganisms? So nomenclature is really just names. How do we name things and how do we put them into different categories or different groups? We're going to learn about different groups of bacteria. There's the gram positives and the gram negatives, and there's other ways to classify them and group them together. And we'll look at a text called Bergey's Manual for the classification of microorganisms. So every microorganism that gets discovered ultimately ends up being classified and put into this group. And how do all these things fit together phylogenetically? Like, how related are they? Is Staphylococcus and Streptococcus more closely related than E. coli and Staphylococcus? Or how are these things related to each other? How are they different from each other? How should they be grouped together? So that's really what a lot of this classification is about. It's kind of how do we put things together? And we'll see that classification, again, it's science, things change. So an example of that is the organism that those of you who already work in healthcare might have heard of, referred to as C. diff. C. diff used to stand for Clostridium difficile. It has been reclassified based on more detailed information using molecular techniques, more modern techniques. It's been changed in its classification, and now it's called Clostridioides difficile. So it still is called C. diff in the short way, but it's no longer in the same genus that it used to be in. It's been moved. For most of us, that doesn't really make any difference. It's still the same organism, but it's a distinction based on differences in its genetic makeup that say, no, it's not really this, it's closer to this. Okay, so that's classification, the orderly arrangement and putting things into a hierarchy, and putting things together that are more closely related. And you'll see various trees and things like that. We'll look at a couple of those as we go through. And then identification, discovering and organisms, and then figuring out what are they and where do they belong in with what we already know. So we have this binomial system that we use for naming microorganisms. And this is true not just for microbiology, but we use it, you will always see things in microbiology named that way. So, for example, you might have already heard of Staphylococcus aureus, and notice how it's written here. It's written italicized. If you were handwriting this, you would underline it. The first word, Staphylococcus, has the capital letter, and that's the genus name. So this is the genus. The genus is Staphylococcus. The species is aureus. So genus name, species name. That's the binomial system for nomenclature. If you were talking about a human, you'd say Homo sapien. Homo is the genus, sapien is our species. Okay? So if you're writing, the first time you would write it out in the long form, and then later in the same, like if you're writing a paragraph or a lab report or something like that, later you could just shorten it to S. aureus. You don't have to write out Staphylococcus every single time. You can just change it to S. aureus, but still it's italicized. The S is capital, the A in aureus is not capitalized. So always follow this nomenclature. It's very important, and in the laboratory, learn how to spell these things. Okay? And the taxonomy. Now, this again, you probably have heard of before. This is going from the largest, so domains. There's 3 domains: bacteria, archaea, and eukarya. We'll look at those in a little bit more detail momentarily. And then we have the kingdoms, phylums or divisions, class, order, family, and then we're down to genus and species. We just saw how in the nomenclature we include genus and species names. So what's a good mnemonic for this? So the one that I think of, and I don't know where it came from, is did King Philip come over for great spaghetti? Okay, so that says, did King Philip come over for great spaghetti? Domain, kingdom, phylum, class, order, family, genus, species, taking us from the biggest down to the smallest level of classification. So let's look at a couple examples of that. So here up at the top here, this is the starting with the kingdom level, and for the domain, we're in the domain of eukarya. Okay, so we're in the domain of eukarya. Here we have Animalia, and you can see some examples there, everything from dogs to humans, etc. And then the phylum, Chordata. These are things that produce a notochord. And then the next level is Mammalia. So notice that as we go down, the groups are getting smaller and smaller and smaller. And of course, this is not an all-inclusive group. This is just a few examples, all the way down to a few examples. And then we get to Mammalia, so things that possess mammary glands and hair. And then we move down to the primates, things that have, the features of primates. And then we get a little further down into the family. And so we're down to just a fewer organisms. And then finally we get down to the genus Homo and the species Sapien, and we have Homo sapiens, so which is a human. That would be us as individuals. But as you go up the chain, we belong to a much bigger group. And then in the same way, you can take it down looking at protozoa. So here's some examples of protozoa, some of the 50,000 or more different types of protozoa. And then we can move that down into the phylum level, class, order, family, and then ultimately genus and species. And here we are with a single type of protozoa. Again, just taking us through that taxonomy. So again, taxonomy is a way to classify organisms, and it was put together by Carl von Linné in the 1700s. It's called Linnaean taxonomy. It was reorganized a little bit in more modern times by Carl Woese when we added the domains, the 3 domains. But it lays down the basic structure and then the way that we name things. And then we have this nomenclature and talk about the branching in the trees. This is where things all started. And then we have bacteria and archaea. Some trees you'll see are organized a little bit different here. This is a little bit up in question still, but beyond the scope of our class. But no question the eukarya came after. These 2 were here first. Eukarya came after, and then all the different organisms that come off of there, these different groups of organisms, and then animals, that's where humans would be. So I wanted to show you this video, but before I show you that, I wanted to point something out to you in your textbook. So when you're reading your textbook, you're going to see these gray bands here. And I'm just going to show you the one video from your textbook in this PowerPoint, but there's lots of them. I won't often put them into our PowerPoint presentations, but I strongly recommend that you look at them. And what's kind of cool, if you're reading the book and you turn on the video, you'll be able to see what that video is. And like after you watch it, it'll stay open like this, and you can relate it back to what you're reading. So if you're somebody that really likes to listen to video, listen while you're reading through and things like that, this can add to it. The book also does have a way to a way that you can have it read the book to you. So if you want to listen to the book instead of just reading the book, or you want to listen as you read along, you do also have that option. I just wanted to point that out really quick because I think sometimes we forget about all these video resources that are built into your textbook. And that's really what makes this textbook a good tool, because it adds those extra layers that you wouldn't have with just a paper book. And I should say, you want to purchase a loose leaf book, you can also do that. That you would have to pay for. The college isn't paying for the loose leaf book, but you can get that. I think it's like $25 or $30, something like that, through McGraw-Hill. That's completely optional. It's up to you if you want to buy that or not. Okay, so let's go back to our PowerPoint. And I just wanted to show us this one video. This comes right out of your textbook.
Living organisms are divided into three major groups: the bacteria, the archaea, and the eukarya. The establishment and organization of these groups, called domains, is based on comparisons of nucleotide sequences of ribosomal RNA. Bacteria and archaea are similar in cell structure and are referred to as prokaryotes. These two groups can be distinguished by a number of important characteristics. All higher organisms, protista, plants, fungi, and animals, share a common cell structure and are referred to as eukaryotes. Eukaryotic cells contain a nuclear membrane, endoplasmic reticulum, and organelles such as mitochondria and chloroplasts, whereas prokaryotic cells do not. In bacteria and archaea, protein synthesis occurs on 70S ribosomes, made up of 30S and 50S subunits, whereas in eukaryotic cells, translation occurs on 80S ribosomes, made up of 40S and 60S subunits. S values are based on the size, shape, and weight of a particle, which accounts for why the sum of subunits of the ribosome does not equal the S value of the intact ribosome. Histone proteins associated with the DNA of eukaryotic cells are generally lacking in bacteria but present in archaea. Archaea histones are similar in structure to eukaryotic histones. Small amounts of protein are associated with maintaining the organization of bacterial DNA. Most bacteria and archaea have a cell wall. The bacterial cell wall contains peptidoglycan as well as proteins, polysaccharides, and lipids. The archaeal cell wall contains no peptidoglycan, but does contain proteins, polysaccharides, and lipids. Cell walls are also present in eukaryotes such as plants, algae, and fungi, but are lacking in animal cells and protozoa. Plant cell walls contain cellulose and lignins, whereas fungal cell walls contain chitin. The cell membranes of bacteria and eukarya contain phospholipids consisting of esters of glycerol, whereas the cell membranes of all archaea contain ethers of glycerol.
So this particular video, I think, has a lot of good information for you, but it's a little bit dense, and some of this information we'll be picking up again in later chapters. But I really think you should bookmark this one, and maybe revisit it when we get to those later chapters, because I like how it makes those distinctions and it kind of explains some of these ideas and concepts of how, that the ribosomes are different, that the cell walls are different, and what those structures are. Okay.
Living organisms are divided into three.
I don't think we need to listen to it twice, though. So as the video talked about, we have these three domains in our universal tree of life, and that is going to be the bacteria, the archaea, and the eukarya. And the bacteria and archaea are of the prokaryotic cell structure, whereas the eukarya are the eukaryotic cell structure. But these two groups, the bacteria and the archaea, are also very distinct from each other, even though they are both prokaryotic. As we saw in that video, the way their linkages are, basic foundational things are different between them. And we can use molecular biology to give better detail. I mentioned earlier that using molecular biology, looking at the sequence of the ribosomes, looking at the actual sequence of the genome itself, the DNA of the genome, in addition to the ribosomal um, structures and things like that, have laid the groundwork for changing the taxonomy of certain organisms and regrouping them, so that even though they might have looked a certain way in the microscope and biochemically they might have behaved a certain way, it turns out that from a molecular point of view, things are related in different ways. So taxonomy can change in that regard. So here's our section check. How are the names assigned and abbreviated? What are the taxonomic categories and the 3 domains? So you can kind of go back through and look at those details. That brings us to the end of chapter 1. So now if you haven't already, you can go into McGraw-Hill Connect, do your readings, do your homework assignment, and then finally take the quiz. All righty. As always, if you have any questions, please send me an email. I'm happy to answer anything that you may have. I will see you soon.